this is the way to go, because now there are no limitations. We can even have meetings hosted from any part of the country, even any part of the world, seamlessly. So basically, today's agenda, we are going to have an opening prayer. Then after the opening prayer, we shall have an opening remarks from the representative from the Ministry of Education and Sports. Then we shall also have an opening remark from the President of the Association of Biology Educators. Then we shall also have an opening remark from a representative from the UICT, then also representative from RENU. Then after, we shall have now presentations from our facilitators, that is Ciano, then followed by teacher Kathy. Then afterwards, we shall have teacher Sheila. Then we shall have responses from teachers everywhere, or even questions. Then we shall have AOB and then the closing prayer. Still amid this task, we expect to get the UNEB biology subject specialist, though he has told me that he's engaged in another meeting right now, but we are hopeful that he is likely also to join. Then if you have some questions, you'll give them, you'll be able to answer. Actually, I've seen he has joined, Mr. Okelo Jimmy, you're most welcome. So he will also come in shortly before the presentations from the facilitators. So without wasting time, I would welcome an opening prayer from a willing participant who is going to give us an opening prayer. Probably if we have a teacher from Ediofi Girls. Now this workshop, though it is online, but it is hosted by Ediofi Girls Secondary School in Arua. So apart from Luciano, I don't know if we have another teacher in attendance from Ediofi Girls to give us an opening prayer. You can raise your hand, the gesture, then I unmute you, then you start us off. Or if he's not there, then let me invite Akira Dennis. Akira Dennis, may you please give us an opening prayer? Akira Dennis. Akira Dennis, are you there? Okay, then we can have Acheng Mary. Acheng Mary, is it possible to get an opening prayer from you?
Hello.
Yes, Mary.
Good afternoon, everyone.
Good afternoon. Kindly introduce yourself, then you give us an opening prayer.
Okay, I'm Acheng Mary. I teach at Kalinavidi. So we can pray.
Thank you.
Heavenly Father, we thank you for this afternoon. We thank you for the people that have organized this workshop. We ask you to come and be with us. May you guide the organizers, the people who are going to take us through. May you also help us to understand whatever we are going to learn and put it into practice. In the name of Jesus Christ, I've prayed.
Thank you so much. Thank you so much, Mary. Okay, I think now with God's blessings, we can know that now the meeting or the session has officially started. Now, my name is teacher Ugonza Arthur. I'm a teacher of biology at St. Mary's College, Chisubi. I'm going to be the moderator of today's session. So with the next item on our agenda is an opening remark from Ronald Dungu, our representative from the Ministry of Education. Mr. Dungu, are you on? Yes, you're on. Kindly give us your opening remarks.
Yes, Mr. Kugonza, I just want to be sure that you can hear me from where I am.
You're very clear.
Okay, thank you, Mr. Kugonza. Thank you, the organizers, for this meeting. I also just wanted to find out if we did not get the head teacher of Ediofe Girls onto the call. Is Mr. Luciano able to confirm that we are not able to find her?
Mr. Luciano, kindly respond to that.
Okay, because we had really wanted the teacher to also be part of the opening remarks, but that is okay if we are not able to get her onto the call. Was that Mr. Luciano? Okay, ladies and gentlemen, my name is Ronald Dungu. I'm a Principal Education Officer in the Ministry of Education, and right now I'm calling in from Karuma. I've just crossed Karuma, coming back from West Nile. And so I want to apologize for the position where I am. And I also requested that I take my remarks before any other person so that I can continue with the journey. I will follow the seminar along the way. But that is also to confirm that you can study from anywhere. I'm under a tree near Karuma, and that means any biology teacher around this place would actually have sat in my position and listened in. I'll also try to follow the seminar all through so that I know whether the teachers along this route can also access us from wherever they are. The simple reason is that technology has come to stay and we must embrace technology. I bring you greetings from Mama, the Minister of Education and Sports. I bring greetings from the Permanent Secretary, who is also out of the country. I bring you greetings from the Commissioner of Government Secondary Schools, and they are all delighted that this is happening because we had a challenge. The challenge was that the performance was not good last year. And if the performance was not good last year, we sit back and look on because we don't have funds, anything, yet close the learning gap amongst the biology teacher through the available technology. So I want to thank the Association of Biology Educators for organizing this event. And I know whichever way it goes, it's already a success because I have been able to connect in. So we can only work towards improving the technology, but the experts are already on the call. That means we can always share when the technologies work. I want to thank all those who have come up as presenters, because that means they are willing to share knowledge. Collaborative learning is what we go, and I know it is the greatest challenge we have amongst our teachers. Teachers do not want to share and wonder how they got the knowledge from someone else, and now they don't want to share the knowledge. So God gave us the chance to live so that we can support each other. And the way to go is really to support each other. I want to thank Renew that is connecting so many schools to the internet through UCC. And I know that the ministry would like to connect over 1,000 schools to the internet. But what is left is, if the school has internet, then what do they use it for? So this is to demonstrate that technology can be used within our schools. And therefore, it is important that this session happens today so that we can have learning lessons going forward. I want to thank UICT. They'll be speaking to us for the training that they've been undertaking to make sure that our teachers embrace technology, and I encourage them to continue the training. I want to thank the Holistic E-Learning platform that is mobilizing all of us and ensuring that these programs go on. We've had about 3 weeks of intensive work, and yesterday we just had a workshop for the head teachers that was conducted by a professor from USA, and all this was to demonstrate that technology has come to stay. You can do quite a number of things with the technology. I want to thank the teachers who are joining in. Please kindly encourage all the other teachers to undertake the technology space so that we can close the learning gap. I know quite a lot of learning is going to happen, both on the technology side, but also on the biology side. Whatever the hiccups, the challenges that may come along the way, let's take them as positive so that we can learn going forward. Thank everyone who is joining in, and I pray have a full discussion. And the ministry, especially the government secondary department, is open to teachers. Please come in and we discuss what is possible, make sure that close the learning gap amongst our teachers, and hence the learning for the learning. Thank you all for listening to me. And at that, I would like to say that this workshop is open, and please carry on. I'll be following along the way. Thank you, sir.
Okay, thank you so much, Mr. Dungu, for that presentation and for also declaring our workshop or seminar open. Indeed, if we embrace technology, then there won't be any limits that would be hindering the closing of the learning gap that is existing currently. So thank you so much for this initiative, and thank you so much for blessing our invitation to participate in this workshop. Thank you so much. So now Idiofi Girls SS is on. Now let us invite the head teacher of Idiofi Girls SS also to give us her opening remarks. Idiofi Girls, they are the host of this year's or this session, this online teachers seminar for this term. So thank you so much. Idiofi Girls, the head teacher, please, could you unmute and talk to the teachers?
Thank you so much, Arthur, and all the participants here. I am called Sister Josephine Achiro Achan, a teacher at St. Mary's Idiofi. We are privileged to host this training today, this workshop. I am here with one of our teachers who is going to be a facilitator, Mr. Lionel Luciano Nsazu, and 3 other teachers of biology of the school. I want to extend my sincere appreciation to Mr. Dungu Ronald, who was here with us this morning at St. Mary's Idiofi, paying us a courtesy visit. We are very privileged to host this biology seminar, but above all, I want to appreciate his initiative of bringing on board all the teachers, all the schools, and specifically using technology to bring us together, to learn from one another, to share the talents, the skills, the good practices that we have in our different schools. St. Mary's Idiofi is a girls' secondary school in Arua City. We have a population of about 1,162 students, with 43 teachers. We are 40 years old. We are young, but we are still growing. We are happy that so far we have been doing well in academics, but that is not yet all. We hope to do better. And one way of improving our performance over time is what we are doing right now, getting to share with others, getting to learn from one another, and bringing our skills, our teaching skills together as teachers of this country. We are privileged that we are hosting this. I want to virtually welcome all of you to St. Mary's Ediofe. I appreciate the organizers of this workshop. And please, this should not be the last time you will be in Ediofe. Come again. We are always ready to welcome you physically here and even virtually. And we are open to this kind of learning. So I want to virtually welcome you to St. Mary's Ediofe. Please feel at St. Mary's. Thank you so much.
Thank you so much, Sister Josephine. St. Mary's Ediofe. What I can say is that St. Mary's Chisuvi is also sending you greetings. And thank you so much for accepting to host and also initiating this kind of interaction that we have witnessed become a reality today. I'm sure this won't be the last one. More other engagements are coming. This now opening the doorways, showing us that actually it is possible to have the whole country gather under one computer or maybe under one platform, and we share ideas on how we can improve the teaching and the learning. Thank you so much. We are very grateful. Now, the next one.
Thank you, Martha. Maybe you can have a look at the team I have here. It will be good for you to see them. I have with me here Mr. Lionel Luciazo, who is going to be a presenter.
Yes, I'm seeing.
I have Madame Felista, a member of the department. Here she is. I have Madame Esther Orishaba, a member of the department. And I have the next person. Yes, this is one of our student teachers on school practice. We are very happy that Mr. Jungu has encouraged us to profile our teachers on school practice. And these ones are already started. They are doing well. They have a mentor who will be accompanying them for the weeks that they'll be here with us for the school practice. Thank you so much. I wish you a good time during this discussion. Thank you.
Okay. Thank you so much, Sister Josephine. Actually, we have ever met once when when doing some work under your name.
Okay.
So research, and since then we've been in touch.
Yeah.
Okay.
Thank you.
Thank you too. Okay, so that is in St. Mary's Ediofe Girls. So now we can have opening remarks from Mr. Dongo Shema. Mr. Dongo Shema is the president of the Association of Biology Educators, one of the partner organizations that has enabled this seminar take place today. Yes, Mr. Dongo Shema, you can talk to the teachers.
Yes, thank you so much, Mr. Asa. I don't know if I'm audible enough. Am I audible enough?
You're very audible.
Okay, sir. I would like to thank the preceding speakers, Mr. Ronald Dungu and Sister Josephine, the head teacher of St. Mary's Ediofe Girls Secondary School, for giving us very encouraging opening remarks for this session, which is bringing so many thinking so many heads into one thinking cap. I would also like to thank the organizations that have extended an invitation to us to partner in ensuring that this webinar is delivered with the best quality of brains. That is the holistic e-learning platform, HELP, RENU, the Ministry of Education and Sports, Government Secondary Education, and the Uganda Institute of Information Technology. My name, Dongo Shema Frederick. I'm a biology chemistry teacher at Natete Muslim High School here in the heart of Kampala. It's a Universal Secondary Education School. And I'm also currently the team leader of the Association of Biology Educators. We are delighted as the Association of Biology Educators to be part of this arrangement because biology is in our hearts. Of all the sciences that we have being offered at secondary at O level, we believe that biology is the one that is closest to the interests of the learners in their daily lives. We therefore find no excuse whatsoever for any underperformance or even poor performance that has manifested over the years. It's therefore our desire that we clear away all the impediments that seem to be causing poor results in biology across the country. The team that we have comprising Mr. Leone Luciano, Arina Itweshila, and Bogere, Arina Itwe Kathy from Mengo Senior School, and Bogere Sheila Lubam of King's College Budo, is a team that we trust. They have the knowledge, the skills that I believe will enrich all of us who are listening in. And I beg that we pay great attention to what they tell us. I also would like to promise that, like Sister has just echoed, this is not going to be the last of such arrangements. We look forward to having several of them as the term runs and as the year progresses, to ensure that teachers from wherever they are, from all the corners of our country, Uganda, who could be delinked by distance from other teachers, make it possible to share the knowledge and the skills that they have gained over time with the rest of us. And we promise that we shall do everything possible to seek for possible answers from whoever seems to have them as to how we can improve biology at both O level and A level. I'm going to be brief because currently we are traveling to Ibanda Secondary School, Western Uganda, for another activity which will take place tomorrow. So we may not be online consistently, but we wish the team and the participants good deliberations. Have a good day.
Okay, thank you so much, Mr. Dongo Shema. We're indeed very grateful. And we wish you a safe travel.
Thank you.
And A-level G seminar.
Thank you.
The next on the agenda, we shall have is opening remarks from the representative of RENU, but nevertheless, amid this task, Mr. Okelo Jimmy, the NEB subject specialist for biology, is also in attendance, and he spoke to us some time back during the reaction. Thank you so much, Mr. Jimmy Okello, for honoring our invitation. We are indeed very grateful.
Thank you. Good afternoon to members.
Good afternoon to you.
Thank you.
Okay. Okay. Now we can have the representative from RENU also introduce himself and talk to us as well. Yes, Timothy, you're on.
Thank you, Arthur. My name is Timothy Ekaru, from the Business Development Unit at RENU. First of all, I want to take this opportunity to thank the other people that have presented, Mr. Dungu, Sister Josephine. I saw Miss Orishaba there. I had an opportunity to meet her sometime back when we were in Jofe Girls. Good to see you there. And when I checked through here, I see there are about 90 other teachers who have joined in. And actually, that's where I want to start from. When we see this happening at RENU, we feel like at least we have a bearing, or we have a bit of a touch that some of the objectives that we stand for are actually coming to reality. I know that there are quite a number of institutions that are with us here already that are actually enjoying the services offered by RENU. And I'm hoping that many other institutions can be in position to join in. So when Mr. Dungu was giving his remarks, he said that right now it is very possible to use technology wherever you are. And he asserted that he was at Karuma from Arua, and he was proceeding to Kampala. And for us at RENU, it gives us great joy that what we stand for to enable research and collaboration amongst institutions is actually becoming a reality. It gives us a lot of joy to that effect. I know very many other members may be wondering that who is this gentleman and who is RENU. So Teacher Arthur Kugonza, if you permit me, I'll just take a few seconds to just give a bit of a preamble of who we are as RENU, where we have come from, and what we represent and where we aim to reach in the next coming times. So RENU is just an acronym. RENU is in full is Research, Education, Network for Uganda. So RENU is a not-for-profit organization. Rather, it's a member-based organization whereby members come together, particularly in the research and education institution. They came together and they came up with an organization that is what we call today RENU. So RENU started way back in 2006. And at the time, it was vice chancellors of universities, CEOs and EDs of research institutions that came together. And they came together because they felt there was a challenge within the education sector and certainly in the research sector. And when they came together in 2006, they met in Entebbe, and the birth of RENU actually started. They brought their ideas together, and at the initial time, they felt like there was no reliable and affordable connectivity of internet at the beginning, right? So they felt like they were not in position to access and collaborate as institutions, yet it was very critical towards achieving some of their set goals. And therefore, RENU came to be birthed. So today, connectivity, at the time when they were starting RENU, they felt like, well, connectivity is the challenge, but at the time it was just a means to one end, meaning that there were other challenges that they had to solve, but they needed to see that connectivity is the first thing that they start on. And today, I'm seeing when we're having these biology seminars, we're in position to do it without any challenges of traveling. We can be in position to do it through the connectivity of internet. And so when Mr. Dungu came and approached us at RENU and said, well, we have a seminar for biology teachers and other seminars for other institutions that are going to take place, and we need you to come in place. We were more than happy to take the opportunity because this is what we stand for. So today we're offering different products and services, all geared and aimed at seeing that the challenges that the everyday teacher faces are eliminated. the challenges that other institutions face. For example, today I'm seeing that Mr. Dongushema had mentioned that they have an association of biology educators and that are perhaps traveling to even under secondary school. So I don't know, maybe I'll just leave my contacts here and then I'll try to get in touch with Mr. Dongu, and even for the purposes of the rest of the people, and we see how best RENU can always come in, especially when you have activities that need our attention. So today, the composition of RENU, I was just giving you just a bit of the beginning of RENU. So RENU was officially incorporated in 2008. And then we got a license to operate from UCC in 2010. And then we had different memorandums of understanding we signed with different other organizations, including NITA Uganda and different organizations, to see that the objective that we set out is achieved. Today, we have almost all the universities in the country connected into RENU. Our aim right now is to see that more institutions, your institutions that you're currently running, the schools, are able to join and benefit from the different services and products that we are running. Teacher Arthur, you will excuse me, I'm not going to be in position to dwell much, but I'll also, if you permit me through you, I'll just leave my contacts here so that if in case there are some queries or challenges that the different teachers want, or they want to know more about RENU, I would be more glad to see that I can be in position to respond to that. I'll end my remarks again by... All right, I will end my remarks by again thanking Mr. Dongo for making this happen. We appreciate you, Mr. Dongo, and I look forward to meeting you tomorrow. I think we'll be having a similar discussion like this. I will be glad to meet you more. Thank you, Teacher Arthur. Thank you, Sister Josephine, for hosting the members right now at St. Mary's Idiofe. And thank you for the rest of the members who are present today. Asante sana.
Okay, thank you so much, Timothy. We're indeed very grateful that the connection gap will be bridged. You can just imagine if actually St. Mary's Idiofe had to host all of us. I don't think that would even have been able to fit or even to travel to Arua for such a discussion. But because of the connectivity, because of the internet, this is very possible. Thank you so much, Timothy. We are waiting. I don't know how you're going to share your contact, but I think you'll share it in the inbox. We shall pick it from there and share it with us. I would like to recognize the presence of Panyango SS biology teachers. You can wave to us. Panyango SS biology teachers, thank you so much. Then also St. Aloysius College. St. Aloysius College, I don't know if you're also video to us. Yes, St. Aloysius College in Nyapea. Thank you. SS Sinjiro, they are also on. Share the video us. Western Uganda, Sinjiro. Then also Gulu Ame SS is also on. Gulu Ame SS biology teachers. Thank you. We shall be honoring some as we move on. Now, we are now ending with about 5 minutes to end our first session, opening remarks. Then after which we shall go to our facilitators. And Sheila will present first. Then we will follow. So let me have a representative from the UICT to address teachers of biology. Yes, the representative from UICT.
Teacher Arthur?
Yes, Mr. Kaziba?
Unfortunately, I'm not seeing Francis on the call, so we can continue.
Okay, thank you. Then now we can now go into our discussions. Now, basically, the main aim for intervening this seminar is to help us get ideas or share ideas on how we can help the current senior 4 students sit and excel in their exams. Remember, we are now finalizing the old curriculum and wouldn't wish our final candidates, the old curriculum, to fail like the ones of last year. So we are hoping that next we can have engagements, we share ideas on what we are doing, how best we can help, then we shall be able at least slightly improve the performance of the biology countrywide. Then we have also incoming online sessions like these ones. We shall have one targeting mainly the new curriculum. Then we shall also have another one mainly for A-level biology. So today it is about senior 4 class. How do we help them to sit and pass their final exams? So Sheila, are you ready with your presentation? You can share your presentation and you start, please.
Thank you so much, Arthur. I recognize the existing protocol as stated by our moderator. My name is Obere Ruvama. I'm a teacher of biology at King's College, Bordeaux. I'm very delighted for this opportunity to share and also to learn from my peers. I thank the organizers and also the participants for making this time. My task, I'd like to call it, was enjoyable because has given something to do with data handling, applying, and evaluation, but at a level of an S4 learner. So usually data questions appear in number 31, and our students usually come, biology would be easy, but number 31. What is in number 31? We usually have experimental setups or numerical data, and that data could be that they're given graphs to draw, or it's an experimental setup and they're supposed to apply an understanding, an interpretation of the experimental procedure. So we need to equip our students with such. If they have data, how do they interpret it? If it involves plotting a graph, how do they plot a graph? They should know why that some certain procedures exist in experiments. You know, like if I'm using sodium hydrogen carbonate in an experiment to deal with photosynthesis, what role is it playing? And then another thing that teases them is a control experiment. How do you determine that this is a control experiment? What is a control experiment anyway? So I hope moving along, these are things that we could handle as teachers or even in this very setup. If we look at numerical data, it's usually, yes, it involves plotting graphs. And interestingly, we've seen it appearing in paper 1 and also in paper 2, in the practical setup. There's some of the graphs could be line graphs. I think our students find them easily. They find them easier to attempt. And with the line graph, it involves just plotting points and then joining them with a free hand. Our students make the error of using, yes, freehand, but then they're not joining points per point, and therefore they get a loose gradient or an erroneous gradient. Otherwise, each point should be, each point should be joined points, at each point per point. Then also, there is a question of histograms. I think we've seen histograms showing up. When do you plot a histogram? And maybe one day we'll have a bar graph showing up. I think this showed up in paper 2, and Kathy dealing with that will handle. If we have it in paper 1, then it's basically looking, if you have non-numerical data or you have grouped data, then you're going to have those bars being plotted, you know. And of course, the interpretation involves biological content. Sometimes when we draw the graphs, if all the graphs are drawn for the students, they could be 2 on the x-axis, not just one line. And the student has to draw using, you know, if you're using a pointer and a circle or a star to indicate, to kind of give a key, a subtle key as to the different variables that are being used on the Y. So yes, interpretation of the data. There are 3 questions that come in. How we tell students, either we have to explain, to describe the graph. Sometimes it's describe the trend of the graph, the changes, the pattern. Other times is to describe the effect. For example, we know that if we say describe the trend, a trend involves a change, and therefore there is a range that is given. You're going to say from one point on the x-axis to another point on the x-axis. What we usually have students find difficulty with, they would like to start with a word initially or at the lowest point. They usually want to make that first observation. Be wrong if the question asks you, you know, to describe the changes. I think we had that last day, describe the changes. And if you have this question, then you're looking at the gradient. You're looking at the gradient and you're going to describe it that, yes, the change is increasing, but you have to also give proof of observation. How is it increasing? Is it increasing gradually? Is it increasing rapidly? Or is it, you know, constant? So there's that distinction that has effect. Apart from exceptional cases, they do not need qualification. An effect is either increasing, it is decreasing, or there is no effect. Yeah, sometimes I laugh with my students when they say the effect is constant, there is no constant or something like that. But yes, an effect is either increasing or there is no effect at all. And if we look at the relationship, if you're dealing with a question that has relationship, then you're going to talk about, you know, you're going to begin as, and you have to appreciate what is the biology. For example, if I'm dealing with transpiration rate and absorption rate in a plant, do I say the relationship is as transpiration rate is increasing, water uptake is increasing, or do I talk about as water uptake is increasing, transpiration rate is also increasing, So when we are looking at a relationship, still we are referring to the content, and then we start with as, you know, as the transpiration rate increases, the rate of water uptake also increases, you know. That is a defined relationship or a described relationship. So that is about graphs. If I could go to graph data interpretation, depends on the action verbs or the question tags within the instructions given. Basically, whether it's a graph you're dealing with, number 31 is a graph or a procedure. I have a simple rule of the thumb I use while I'm instructing students on question approach. If somebody has a pen, if you have a pen with you, you could write this could I call it an acronym? You could write THANKS. I use that with my students. That is THANKS, and it will summarize all this that we are looking at. The T is for the topic, yeah? I tell them, when you're dealing with number 31 that you're fearing so much, identify the topic therein. The topic is going to give them the keywords, because if they identify the topic is movements in and out of cells, and they're going to talk about diffusion, that means they're going to have the keywords of movements along a concentration gradient. You know, if they identify the topic as respiration, then they'll be talking about energy. Maybe they'll look at aerobic respiration. Is it anaerobic? So identifying the topic is very key as they're reading the title. No, sorry, the introduction of the question. As they're reading the introduction of the question, whether it's leading them to a graph or it's leading them to data, just data in a table or an experiment. Yeah. And like I said, the topic leads to the keywords. So that care within what we've written is actually to give you the keywords. If you're talking about osmosis, yes, we could use words like isotonic, hypotonic, hypertonic. Yeah, you'll find that those will be used. In fact, I tell my students, the stem of the question actually has the answers. When I read, there's a question that came of zebras and lions, and they talked about an isolated island. Yeah, I expect a student to underline that word isolated, interpret it. Yeah, what does isolated mean? So if with identifying that word, a student could easily realize that, okay, there's no interaction of other animals apart from lions and zebras. When we go to the next one, the H is for the hidden information. Yeah. If there's zebras and lions and they're together on an island, on an isolated island, one is going to be prey to the other. One is going to be a predator to the other. And that is the hidden information. And I tell them, actually, and you know, that's why I tell them that actually the question has the answers. And in that question, if you go back to that question, that is a past paper question, when you go back, you realize, yes, actually, if a student identified the hidden content, yeah. If I talk about that somebody had a meal of carbohydrates, and later talk about glucose absorption, then the hidden information is about digestion of the carbohydrates broken down to glucose. When we come to the question tags, yes, they're within the question, and I make it an A and say there is an action that is being given. Yeah, when you look at these questions, are they telling you to compare? And when they say compare, there you're giving differences and similarities. How you state them is also important. Students get wary of stating a difference in full, and they stop somewhere. You know, respiration involves you know, use of oxygen. Photosynthesis does not, something like that. They shouldn't get wary. So the stating of the differences is also important. But we are going back to the action that is given. When they tell you to describe, what are you doing? Of course, you're using a descriptive word therein. Yeah. Then if they tell you to explain, you're giving a reason. But if they say explain how, that action is usually pointing to a mechanism. Explain how. Yeah. If the action is to suggest or outline, now that is a simpler task, you know, that is more easier to attend. So there is need for students to understand the question tags or the action verbs that are involved in the question. The N, I use the word nuance, but I soften it to tell students that these questions have principles that they work with. Principles, if I'm dealing with gaseous exchange, definitely I'm dealing with diffusion, yeah? Because everything about gas exchange is about, you know, oh, where is oxygen going? Where is carbon dioxide going? And it's along a concentration gradient. Whether I'm talking about the respiratory surface and its adaptations, I'm still looking at the, the factors that affect the rate of diffusion. If I'm looking at gaseous exchange in any of the organisms our syllabus specifies, it's still about diffusion. When we go to the other side of respiration, the principle is actually energy. So whenever somebody talks about respiration, they're going to be talking about production of energy, whether it's aerobic or anaerobic. So with each topic, the task is to the student and also to the teacher to say, what is the principle? What are the subtle or the distinctive and precise pointers of this topic, that when a student identifies it in the stem of the question or in the introduction of the question, is able to go ahead. If we say photosynthesis, really the factor behind it is light. And therefore, if somewhere they say put the plant in a dark place, yeah, already the student can point out the hidden information is in the dark. The topic is photosynthesis. The principle is that, photosynthesis occurs under light conditions. So during graphs, I don't know if if the graph was representing maybe an experiment, there could have been need to look at, they could be need to look at the process, how it's, where it starts from and where it ends. Even if you're looking at the various experiments, for example, if you're looking at germination, it's somebody sets number 31 in germination. We know the first step in germination is about imbibition, and without that, a student may be denied marks. So yes, the sequence is also important. So with question approach number 31, I've summarized all this in that question approach. I don't know if somebody has recognized the word that we have actually written. If I could mute and somebody offers the words that, word that we have written to help with in question approach.
Sheila, it may not be possible to control the background noise. Members are muted.
The question I have is to see if my class was following. Have we recognized the word that we've written to help with question approach? Somebody to offer.
Maybe the members can use the reactions and they put the sign with the man with the hand up so that we can be able to unmute you and you respond. We have Mary.
Hello, thank you so much, Arthur. I did not follow from A. The word is thanks, right? Madam Sheila?
Yes, Mary, the word is thanks, yes.
I understood up to action verbs. The N and K and S, I didn't understand so well.
Okay, I can repeat that. If I can proceed and repeat that.
Thank you. Thank you.
So repeating the word thanks. Okay, I'll repeat that. Members, slightly, I know we have time to consider, but let me take you back to the N. N is an English word. A nuance gives the precise, it's about, when somebody talks about a nuance, they're referring to something that has precise meaning, precise expression, precise attachment. I found that, yeah, if I use principle, it's better. Like, what is the principle? So the N, you can put there P, but, or you can know that it means the N is there for P. There is a nuance for each topic. Look at whatever topic, yeah? If we look, like the examples I used, if you look at respiration, you cannot talk about respiration and isolate energy production from respiration. Well, if I talk about gas exchange, I cannot isolate the principle of diffusion from gas exchange. So when students bring their question and you help them identify the principle, they're actually able to go on and answer the question by themselves. You know, it kind of takes them to where is the starting point. Yes, you've read the question, you have identified, yeah, the topic. If I look at the topic and I go to the subtopic or the sub-theme in the topic, and it's germination, yeah. The first, what is about germination? What is the principle behind germination? Something like that. So when you're teaching your students, you can develop with them, for example, there are even certain words we can use, and like if a question, you identify that it's talking about size, where do you go there? From there, if it's talking about size, then you go surface area to volume ratio. So that is the nuance. That's the nuance. Or if you're talking about heat loss, heat gain, what is the principle behind that? Something like that. You can develop it together with your students as you teach topic by topic. You help them that whenever I reach this topic, this is the riding theme. This is the recurring theme in this topic. This is the recurring biological principle. Then with K, I was referring to the keywords. Again, there are words that need to go with an explanation in the topic you're dealing with. If you're dealing with coordination, then stimulus is something that goes with that. That may have to appear in the answer. When you're dealing with osmosis, the concentration, the differences in the concentration is something that you have to deal with, or the concentration gradient and the type of molecules. So there are those keywords that you're like, okay, if you're dealing with osmosis, these are the keywords that come out. And also it helps them identify when they read the question, they should underline the keywords within the question. And keywords, sometimes I say those keywords are the ones with biological significance. I don't expect a student to underline of, in, under. Yeah? Sometimes they don't have biological significance. Again, I say it in exceptional cases. Somebody could say a plant, you know, an aquatic plant found on water. Now on becomes important. That means it's non-submerged, it is floating. But you know, those are at a higher degree. Normally the
Sheila,
keywords, yes?
You have the question from Wilfred Sentongo. Okay, he's mute. Akela Denish, you have something to ask?
You say, good afternoon to everyone. Yeah. My question is little. I want to know how dependent variable can also appear too on the x-axis. That is my question.
How dependent variables can appear on
two, because from if you check your slide, number 2 or number 3 is there is some note there that take note dependent variable can also appear too on the x-axis. That is the question.
Okay,
I want
Sheila, you can talk about that.
Yeah.
Okay, so let me
much.
deliver what we're talking about with Mary. And lastly, we have sequence. The S is for sequence, and we're saying that one will follow a certain. There's, you tell your students, you bring it to the student, that some reactions really have a beginning point and an end. You can't start in the middle. That is important. So that is our question approach. The last question, the previous question we've had is, when you're dealing with graphs, you're really dealing with a why, with the dependent variables. If you realize, that's why our title is usually a graph, not usually, it's always a graph of y, against x, it's because you're looking at what is happening on the dependent, on the y-axis. So how can you have two? Sometimes if I give you an example of sweating and rate of urination, rate of sweating and rate of urination, they can be together. Yeah? You have just temperature, as when you look, the temperature is increasing, sweating, the rate of sweating will increase, but the rate of urination will decrease. So that is how they can appear too. That's what we are referring to. Have I answered your questions?
Yes, I think, Sheila, yes, you can proceed.
Okay, so I'm on to my last slide, and this one we already covered when we said action verbs or question tags. That's when they say state, these ones are just talking, this is simple recall. It's right there with the word out to describe, they're telling you to use, descriptive language. And when they say explain, that they're looking for you to give reasons or two, reasons that relate to a scientific concept, or if they add on explain how, that is to give the mechanism. Here we have anatomy, descriptive word. Yes, sometimes you have to relate structure to function. And the way we question our students, we ask them to give the adaptation. And of course, question approach, you say, relate structure to function. But it's not just enough to relate structure to function. Yeah, for example, if I say, how is a leaf adapted to photosynthesis? It's not just enough for a student to say lamina to absorb sunlight. That is not an adaptation. When you come to the structure, there should be proof of observation that a student has ever seen a leaf and appreciated the structure of the leaf. If I say a broad lamina, I've given proof of observation. Then I must qualify. Why have I used the word broad? Qualify, yeah, the adjectives chosen. An adjective is the descriptive word we attach to the structure, to the noun. So if I say broad lamina, why have I used the word broad? To increase the surface area, and so forth. Of course, there are times when the student may not be able to use that. For example, if we say a leaf has chlorophyll, that one is definitely to absorb sunlight and such absorption of sunlight, but you indulge them. So if there are no more questions, I'd like to submit my discussion. I thank you for listening.
Okay, thank you so much, Sheila, for that elaborate presentation.
Over to you, Arthur.
Of course, there will be some questions, but I think we shall entertain the questions when all our three facilitators have finished, because we may end up asking things that are going to be presented by the other remaining two facilitators. So kindly, I pray that we hold the question firing session to the end. Thank you so much, Sheila. Okay, now that is Sheila Wogere Rugwama. Now, our next presentation or our next facilitator is none other than Mr. Luciano from St. Mary's Ediofi Girls SS, our hosting school. So Mr. Luciano, kindly unmute and present or talk to the teachers. Thank you so much.
Thank you, Arthur. I want to thank the...
Sheila, you can stop the sharing.
Hello? Hello?
Yes, Luciano, we are with you. Bring it clear.
Right. I want to thank the initiator of this program for bringing the whole country together under one umbrella. I think that in the past wasn't possible. I'm very grateful for this initiative. We are here trying to address an issue that has been making headlines of recent, and that is the poor performance in biology. Biology, as already commented on by other people, is at the very heart of the sciences. And so when we see our learners not doing very well, it is so disturbing. And our prayer is that as we conclude this all the curriculum, where the last set of learners are right now in Senior 4, we should conclude it with a positive record, a record that would live to remember. And so I have a few issues, a few observations, experiences, ideas that I am going to share with my audience. There could be obvious observations, there could be obvious points, but it is very good that we try to remind ourselves of these obvious. I'm going to handle tips on how to answer Biology 553/1. I happen to have had a good interaction with this paper for some time. And from all that I have experienced, I have gathered a few information which I wish to share with you. When you want to skin an animal, it is very important that you first get to understand the structure of this animal so that you know where to first start laying the knife to open the skin. Knowledge and understanding, many times, many people have confused them, the two words. They think that knowledge and understanding are synonymous, but when it comes to making sense of them, knowledge is a little different from understanding. I am still waiting for my IT man to come and help me in sharing the screen as I continue. Knowledge, in the way I understand it, is an accumulated body of facts, skills, information, creating an awareness that one gets through experiencing whatever happens around. So it is a form of awareness or familiarity. That is all we can say about knowledge. And as for understanding, understanding is at a higher level. Understanding is at a higher level, and it is the ability to comprehend or articulate facts, skills, information, and awareness gained through experience. So we start at a low level by gaining skills, facts as knowledge. And when it comes to comprehending it and getting more to understand about what you know, then that comes at a little higher level. Structure of biology paper 1. For us science biology teachers that have been handling this paper, it is a 3-in-1 paper, that has 3 sections. Section A has 30 multiple choice questions marked out of 30 marks. Each number has a mark. Section B has 3 structured questions which require filling in of marks, rather points. My sister Sheila elaborately dwelt on question number 31, which is a little bit extended, marked out of 20 marks. I remember the year this number was introduced to this paper. It was this time that performance in biology started going down the drain. Earlier on, we would see our learners, performing very well, scoring very high marks, and raising the faces of many of us in our schools. But from the year question number 31 was introduced, I started seeing a downward trend in the performance of biology, mainly because that number 31, many of our learners seem not to have come to terms with it. And I want to thank Madam Sheila for elaborately taking us through. And it is my serious belief that those of us who are part of this audience in this arrangement will be able to take this down to our schools so that this effort is not in vain. We have question number 32 and question number 33, both of which are marked out of 10 marks each. And so section B has a total of, 40 marks. When it comes to section C has 4 essay numbers, out of which our learners answer any 2 that are to their best test. And each of these 2 numbers is marked out of 15. And therefore that gives us a total of 30 marks for section C. Let us note this. We have 30 numbers in section A, 3 numbers in section B, and 4 numbers in section C. The number of questions from each of the content areas, I mean the topics or the chapters, so much depend on the following considerations. One is the relative importance of the content area. You're all aware that of late, there is a lot of environmental campaigning going on. And if you sample our past papers very carefully, you'll see that there are a number of questions that are picked from the areas that address issues to do with the environment. Then another area is the width of the content area. Some topics are so short, others are long, extended. And where they are very short, you don't expect so many questions to be picked from that area. And where they are so long, so extended, we expect an equivalent number of questions possibly to be coming from that area. Depth of the content area has the same regard. Finally, the expected behavioral outcomes. What do we expect the learners to turn into after experiencing such a learning outcome. And since we are here looking at raising children that will go out to manage our societal challenges, we try to look at such areas that we see really apply to the practical sense of the lives of our learners after the classroom experience. Particularly talking about section A now, a clarity that I want to make is multiple choice questions are not necessarily the same as structured questions or objective questions. Objective questions are structured, objective meaning that the answers are definite. But when we talk about multiple choice questions are part of the objective questions. So particularly addressing this first section of this paper, we try to briefly go to look at what is there about the questions that make up this section. Like I said before, the questions are picked from all the topic areas. I was sampling the topics, and I think from what I saw, we have about 31 topics O level. Yet this section has 30 numbers. So as we prepare our learners, let us encourage them to, as much as possible, move through or go through all the 31 topic areas so that they may not miss out on a fact about whatever may be coming from a particular topic area. Multiple choice questions are questions that have 4 alternative answers made up of 3 parts. And these 3 parts are the stem, the distractors, and the key. The stem is that which sets up the task for the learner, for the test taker. It must always be very clear, and we always make sure that they are very clear. In your settings within the schools, even the enabled settings, I think the setters have always made sure that the stem is clear. The task to respond to is very definite, so that when it comes to making choice of the correct answer, which is the key, the distractors don't seem to be so similar to the key. So of the 4 answers, 3 are very plausible. They may not be correct, or they may be correct, but they may be less correct than what we refer to as the key. And therefore, each number will always have 1 key, the correct answer. And the letter that represents that key must be clearly and visibly written in the box provided on the right-hand side of that question. In a situation where a learner or a test taker changes his mind or her mind and feels that the first choice wasn't the correct one, we kindly advise that such a test taker crosses out that wrong letter neatly and smartly, and then puts the next correct alternative answer outside the box on the right-hand side so that it is very visible to the examiner. We move to section B, the structured questions. Question number 31, Madam Sheila, thank you very much. You already handled this, so I'm not going to talk about that again. All I want to emphasize is for us teachers, the teachers of biology, this number 31 seems to be a thorn in our flesh. Please, let's try to get down to our schools and put into use all the guidance that Madam Sheila gave so that this year we can turn the events around. Continuing with the other questions in section B, number 32 and number 33, I again want to say that these questions are picked from any part of the syllabus, anywhere. It could be the first topic, it could be from the second topic, the last topic, we don't know. But the questions will be spread such that no particular area is over-assessed. And these questions are subsectioned. They are subsectioned in such a manner that maybe we can have part A, we have section B, we have section what, in the order that the first subsection or sub-question will always try to test knowledge. It tests knowledge. It could be asking for definition. It could be asking for listing of factors affecting some kind of process. And this does not require a lot of cognitive involvement. It is just recall, it's knowledge-based, and the marks allotted there are normally equivalent to the level of demand. And as you go to sub-question B, subsection C, question C and D, the strength and the demand of the tasks keep increasing progressively. So they turn it towards the HOTS, high order technique questions that demand a lot more of thinking and reasoning. And the marks allocated thereafter to each one of those will therefore be raised accordingly. The blank spaces left to fill in these answers are believed to be sufficient for the expected responses. In my experience, in my interactions, I have discovered there are some learners that think that those spaces left are too small, so they subdivide those gaps. For example, you have left 2 lines, but the learner will further divide the 2 lines into four other lines, and therefore squeezing the writing and reducing the handwriting to sizes that are not clearly readable to the examiner. And this can become a disadvantage to this learner. And so when we get back there to our schools, let us tell them that the people, the item writers, the people who set these questions always know that the space that they have left there is enough to accommodate the expected response. And therefore, let's encourage them as much as possible to limit their answers within the provisions left for filling in those marks. And therefore, these answers should be short and concise, complete sentences. We discourage learners repeating the questions in those spaces, because if they do, then they take up the space and they will have nowhere for the answers to land. And therefore, please, let's emphasize this. Straightforward answers, short, concise, complete sentences, no repetition of questions in those areas. And therefore, we are saying there are normally no 2 questions from the same area in this section. If question number 31 maybe is from the area of ecology, maybe section B, sorry, the question number 32 can be coming from maybe classification, and question number 3 from another area. It is a huge error if this section has 2 numbers coming from the same area. Section C, the essay questions, Madam Sheila tried to touch a little on how to progress on responding to the tasks in this kind of questions. But I want to say here that there are 4 questions picked from different topics, any part of the syllabus, of which only 2 are attempted by the examinee. Just like for questions in section B, the questions are subsectioned, A, B, C, and so on and so on. And the levels of demand will progress as the numbering also progresses. That means part A could be about definition of a process. It could be about just recalling facts, and it doesn't require so much thinking. But as you progress to other sections of this number, the demand level increases. So the questions start with the lots, low order technique questions like definitions, stating, conditions, and so on, and ending up with hot descriptions and explanations. Marks to each sub-question indicated on adjacent side reflect the level of demand. Sometimes the children overwrite and sometimes they underwrite. in order for them to be able to project the amount of writing expected of them responding to the tasks, it is advisable that we get to tell them that the amount of marks allocated there is normally equivalent to the level of demand. So no questions are subjective. They don't ask for opinions. They are definite. And somebody at one time made a joke, cracked a joke that we teach history of sciences. Many of these principles and facts were discovered long ago. We only try to recapitulate them, we try to propagate them, except that the application may vary a little bit, because two of us can have the same idea, but when it comes to using that idea, the use might take different dimensions. And each of these questions has a maximum of 15 marks. Now, the 4 numbers given, there are children who sometimes think that the 2.5 hours is too much for them. After doing the first 2 numbers, they go for the 3rd one, they go for the 4th one, and end up answering all the 4 numbers. But I think nationally, there could be an agreement, I am speculating, that because the rubric says answer only 2, I am sure the examiners are only obliged to answer the first 2 numbers and they ignore the other 2, because the instruction says answer 2. But the disadvantage of this is if like the 3rd number and the 4th number attempted would have scored the highest marks, and the first 2 attempted scoring, you see, the lowest marks, this learner has disadvantaged himself or herself. And therefore, let's emphasize the fact that when they're making their choices, out of the 4, let them choose the best 2, the 2 that they think that they are able to write with the best trust. If they trust, that should be the area that they can harvest marks from. Now, answering tips. How do we answer? 1, let's emphasize the learners following the rubrics. The rubrics are the instructions. Every section has an instruction. There are those general instructions, but each section has some particular instruction. And therefore, let us ask our learners to familiarize with the action verbs in those questions, in the stems of the questions. For example, if we are defining what are we doing? If we're stating, what are we doing? If we're listing, what is the learner expected to do? If we are describing, we are explaining, we are comparing, what is the learner expected to do? Some of these words, Madam Shell already gave an explanation on. Number 2, for a question requiring description of an experiment, there must appear the following in the learner's work. For example, the question can be asking you to describe an experiment to show that a green leaf produces food by photosynthesis. Or it could be that sunlight intensity affects the rate of photosynthesis in an Enoloidea or in aquatic plant, varying the distances between the plant and then the source of light. When it comes to describing an experiment, we're saying the following facts must appear. They must be consistently arranged. And the first fact that must appear there is the title to the description, to the experiment. The title written in a full sentence and in very correct English, boldly written up there either in capital letters or in small letters, but must be underlined so that it can be separated from the rest of the literature that will follow. Title is number 1. Number 2 is the learner must be able to list the requirements The requirements include the apparatus, some of which are conventional, others are improvised locally, they're locally made, and then the materials. Let this learner list all the required items for this particular experiment, although he or she is handling it descriptively, not practically. Then after that, we could ask the learners to have a diagrammatic representation of the would-be process, giving an impression of what it will be like if this learner really arranged the thing, the apparatus, the materials, arranged them to let the process work out. What will it look like? So the diagrammatic illustrations are very key here. But sometimes whether the diagrammatic illustrations can be scoring marks or not will depend on the framing of the task. There are questions where they will say, with the help of labeled diagrams, describe, you can continue, with the help of labeled diagrams, describe this and that. But there are some questions where they'll just say, describe an experiment to show that this is. So if you bring in the issue of the diagrammatic illustration, maybe that's a bonus for you. One extra mark makes a difference in the life of this learner. So whether called for or not, let us encourage the children, having this diagrammatic impression of the would-be apparatus arranged to work out this kind of experiment. Next is the description of the experiment must be very systematic. It should all start from you picking the first apparatus material and then fitting it to the other one. And it progresses like that until when you have made mention of the use of all the apparatus and materials. Because if up there, under the list of requirements, you have mentioned, let us say, 10 of these requirements, and in the description of the experiment, you have only used 5. Chances are high that you have lost some marks along the way because you have not made full use of the apparatus that you mobilized. And assuming that these apparatus are really the relevant ones for this experiment, leaving them out in the description may be a sign that you have missed out certain concepts that would be earning you some marks. So please make sure that the consistency is maintained. The flow must be undoubted. And at the end of it all, remember all scientific experiments are timed. They are timed. Do the timing correctly. Any description that lacks time will affect your scores in the other concepts that are following this. And if your timing is wrong, for example, you are looking at an experiment where plant leaves lose water by transpiration. I think 3 hours would be enough timing. But where you're going to take it to 12 hours, 24 hours, I think that becomes unrealistic. So in this case, the timing is wrong. Or if you say this experiment is only going to stand for, let us say 10 minutes, again, that timing is very unrealistic. And what will be the consequence of this? The consequence of this is that the marks for observation and conclusion might be affected. So ensure that the learners do the timing appropriately. Correct timing must be emphasized for each of the experiments that we know about that are there in our syllabus. Some of these experiments have control experiments. They have control experiments. And you would not expect that the real experimental setup you describe, I don't know how many words you're using, maybe 100 words, 200 words, and then you think that you describe the control experiment the same way. I just want to advise you, it looks like for the control experiment, this learner, even if it produces an extended writing, this learner will just be given 1 mark. So what am I saying? I'm saying the real experiment, do the description consistently in a very smooth flow
from the starting point up to the last point, timing, everything. But at the end of it all, you only make one statement to cover the control experiment. And in that statement covering the control experiment, you now bring in the factor that would have effect on the success of that experiment. So it becomes the control experiment, and you only get 1 mark there. So let us emphasize giving accurate and elaborate description of the main experiment, and then about the control experiment, one inclusive statement would be enough, because there we might only send 1 mark there. We might only allocate 1 mark. Otherwise, if there is no timing, there is no control experiment, or timing is wrong, chances are very high that this learner is going to lose marks for observation and conclusion. Marks for conclusion and observation will be affected by timing and also presence or absence of the control experiment. That is a very serious thing for us to note. Such questions are very common on our papers, and when we get back there, please let us take this information home. The observation must be stated in line with the title or the aim of the experiment. When you're observing, remember you started from somewhere. You set for yourself an aim. And now the procedure that you follow, the description that you gave an account of, is trying to guide you towards achieving that aim, which becomes your title. So when you're making your observation and the conclusion, remind the learners to keep in mind that there is an aim somewhere that they have to tag their conclusion and observation to. Point number 3 that we should note when we are guiding our learners to answer the questions is that when we're describing processes, scientific processes, for example, maybe we're describing digestion of, let us say, protein, digestion of the carbohydrates. No, we should ensure consistency. For example, when we're describing the digestion of protein, what happens in the mouth? Look at all that happens in the mouth, then go down to all that happens in the stomach, then from there go down to all that happens in the duodenum and the ileum. A learner might start, sometimes they confuse the literature so much. A learner might start from what happens in the mouth and they forget something, goes to the stomach, what happens in the stomach, and then he remembers that I should have mentioned something about an occurrence in the mouth, and then brings in that forgotten item. You see, the flow will have already been violated. And what I know is where consistency has been breached, the marking also stops there. The marking stops there, even if the other information flowing thereafter are all correct. Marking is going to stop there. Let us emphasize consistency and flow so that we don't mark backwards. Marking must always be in progression and in forward direction. Otherwise, marking stops, as we said, when consistency is messed up. Avoid the use of non-biological words or slangs or unnecessary expressions in the description. There was a year, the children were supposed to boil leaves, to describe an experiment to show that plant leaves produce starch by photosynthesis. Although we are aware the food produced is not starch, but eventually the food produced is converted into starch and stored. So testing and finding starch present in a leaf indicates that a process of photosynthesis at one time took place in this leaf. So what would they say? They would say, you simmer. Simmering. Simmering is a slang. We don't have to use words like that. In the essays, there are children who will say, not only that, but also No, these are unnecessary expressions. They waste your time. They waste the space. They waste the examination material. Be biological, write what is there the way it is supposed to be, because in biology, we emphasize objectivity. There is no subjectivity. If you're going to write like you are writing history, for those other extra expressions, most likely you will have wasted your time and you know earn any marks. Finally, but not least, following the rubrics, we are continuing with them. When asked to state or list a certain number of facts, for example, they say list three factors that affect the rate of photosynthesis. 3 factors. When you go to the 4th one, the 5th one, the 6th one, you'll have violated the rubric, because the instruction says only 3. And what also is very painful is those who go beyond the required number. The first 2 or 3 factors may be wrong, or answers may be wrong. And the correct answers begin from the 4th one and they continue like that, you see. Now, you can see the examiner is stopping with the marking on the last number as required by the instruction. But for this learner, the correct answer started from the 4th point, and you can see it is so unfair. And so let us tell them to be very confident and always follow instructions so that they may not misfire at all. Only the official or stated number of questions must be attempted and must be one's best 2 in section C. I think I talked about that already. Use same ink color of the same intensity writing to avoid being suspected or for malpractice or for collusion and so on. Encourage the candidates to write in very clear handwriting. Many of our examiners are already in their years of conclusion, and I seem to see many of them are even now putting on glasses, and so chances are high that where a candidate is not writing very clearly, this examiner may not read through the learners or the test takers' work with the concentration expected. So in order that the learners don't unfairly lose such marks, let's encourage them to write very clearly. Otherwise, this is all I had time for. And I want to thank you. I submit.
Okay, thank you so much, Mr. Luciano Alionzi, for that presentation. And thank you for now equipping us to actually, when we invite facilitators, stand in front of our candidates. We shall also process, make sure it is complete. If you interfere with the process, marking stops there, so that these learners now know. So thank you so much, Mr. Ciano. Of course, you'll have some questions to respond to, but that one will come after all our presentations. Now, I wanted to Mr. Kelojima, as if he has left. Mr. Kelojima, could you still be in? I wanted Mr. Kelojima to come and clarify some things, to answer some questions we have, because he had told me he had some engagement, another meeting. Yes, okay. Okay, now, Mr. Kelo, one of the burning issues I always find when I interact with the teachers is the assurance on the abridged curriculum. People don't know or are still confused. So as you name, what do you have to talk about this abridged curriculum, Mr. Kelojime? And also, as we are guiding our learners or our candidates in these final sessions, what exactly should we do? Mr. Kello. Could we have lost him again? Okay, so we seem to have lost Mr. Kello Jimmy. But we hope he checks in again. Okay, so meanwhile, let us now go to our final presenter or facilitator, the one going to handle one of the hot issues, that is now the practicals. We are aware of the challenges in some of the schools in terms of having access to good laboratories or even having the resources for the practicals. And surely some of the students are either going to do practicals for the first time in the mocks, or they are going to do these practicals for the first time next term when they're interacting with your neighbor. But nevertheless, as teachers, at least we can have this theoretical background that we can now know when we stand in front of these students to brief them about the practicals. What exactly should we tell them? How should we guide them? What are the don'ts and what are the do's? And what are those things that we are supposed to have taught these students? And should you have missed anything, then kind of you pick it up from the presentation, you go and re-emphasize it with your students. So you're welcome, Madam Kathy. Kindly, you can now talk to the biology teachers. Thank you so much.
Thank you very much. Good afternoon, everybody. I take the privilege to present to you prerequisite experimental skills and knowledge, experimental skills, knowledge, and investigations of biology. Just like Arthur said, I'm by the names of Arenaito Catherine. My outline will just be as that one. I'll have a background. I'll have the need for excellence in exams, composition of the practical paper, question tags or question approach skills according to practicals, common mistakes made by students, setting skills for theory and practical questions. My background. Biology practical mainly entails activities that demonstrate biological concepts, and therefore enabling students to apply their knowledge and skills. Tips on how to pass biology. Mainly, we look at the review of the instructions carefully. A student should be able to read instructions before starting to make sure that they understand what they are required of. Then we also have time management. Management of time is also a crucial skill that is supposed to bring out the outcomes of a particular objective or experimental skill. Still, in instilling biology practical skills in learners, we have certain skills that learners should be able to put forward. For example, they should be able to record data. In an experiment, for example, they can be able to record data, like in a soil experiment, which we are looking at the rate at which drainage occurs in particular soil sample. Then they should also be having critical analysis and interpretation of that particular data they've recorded.
Then from there, they should be able to interpret the part of content and apply it in problem solving. For example, if we have soil samples, we have sand and clay, how can we apply content of what they have learned in class in this practical situation? That I have 2 soil samples, how do I improve these soil samples to be fitting in a situation where we have to have high crop yields? So a learner should be able to bring out that problem solving issue. Then should also have critical observation, critical thinking, as well as drawing skills, recording of observable features. They've been given, for example, bones. Are they able to bring out the features that enable that bone to carry out its particular function? How is it adapted to its function, use of those features? And you remember Sheila talked about descriptive words. Every time you talk about a particular characteristic, it should be escorted by a descriptive word. Relating these structures of organisms to their functions, that one goes down to the word adaptation. Do students know and understand the depth at which they should interpret the word adaptation? We need a structure. You identify a structure, tie it to a descriptive word, and later relate it to a particular function. It is crucial to know what is examined, how it is examined, how to answer, and plan for your answer. Before a student is able to attempt a question, they should know the skill of planning. And it is through this planning that they will be able to bring out the concept. They will be able to be precise and specific on certain issues. Bloom's taxonomy. We normally apply Bloom's taxonomy to bring out the different levels of thinking, which include knowledge, that is simple recall or remembering a certain part of a concept. Comprehension. Students should be able to understand the simple recall and relate it to a particular level of thinking. Understand what exactly has been put forward. Then application, how information is applied to solve a challenge. Analysis, Mr. Luciano has really brought out the high order thinking very well. I don't need to really drill on this. Then we also have evaluation. How do we create synthesis in a certain situation to bring out a particular way in which a part of content can be synthesized and then apply different analysis to bring out particular type of information. The need for excellence in exams. Why do we need excellence in exams anyway? Excellence is the measure by which how well a learner performs in exams. And therefore, there is a need for excellence, or the need for excellence is to contribute to good grades, to qualify for biological courses, to gain a wider career opportunity, and avoid high competition for jobs. Application of lab investigations, practicals. Why do we need practicals? Why do we study practicals? To enable the use of techniques and equipment, to enable correlate observations with known facts. These known facts are normally coming from the theory side. We normally want to merge the theory side with an observation, something that the student has seen actually in the lab. Investigations to enable investigate concepts presented in information. For example, osmosis. We study osmosis in class, but is this student able to investigate it in a practical format and apply the concept of osmosis, explain the phenomenon? Composition of the practical paper. Practical paper is mainly composed of, we can examine in physiology, chemicals of life, enzyme activity, that is the most common enzymes, is enzyme catalase and amylase. We have cell physiology, how the cell works. We can look at osmosis or diffusion. We have soil components, looking at the different types of soil, the characteristics, How can they be improved for crop production, etc. Zoology, animal structures, their adaptations and drawings. Students should be able to display good drawing skills, understand the adaptations of these different structures. And we said, when you identify a structure, you should be able to tag it to a descriptive word. Those are tied together. They are not separated. Then we have botany, plant structures, adaptations, and drawings. Now we go into the detail of chemicals of life. Chemicals of life, normally we look at food tests. Is a student able to identify a food substance? Can they write the correct observations and correlate it to the correct deduction or conclusion? What quantifications are required by a student? When I say quantification, yes, a student has identified a food substance contains starch, but how much starch is present in this particular food substance? We can have blue-black that can correlate with moderate amount of starch present. We can have pale blue-black that can correlate with little amount of starch present. We can have black, plain black, normally found in plant storage organs, like pouring iodine on Irish potato. It will completely give you a black marking, showing that this Irish potato contains a lot of starch. Then we have enzyme activity. Enzyme activity, normally we look at the common enzymes. Enzyme amylase. Amylase is a common enzyme that is known for starch breakdown. We can look at effect of temperature on enzyme activity, effect of pH on enzyme activity. What are the most probable temperatures at which an enzyme is going to work best? Every 10 degrees increase in temperature is going to increase the activity of this active substance. However, there is the optimum, 37 to 40 degrees centigrade, where the enzyme is going to give us maximum output, maximum enzyme activity, and this is referred to as the temperature at which the enzyme works best. We have effect of pH. Enzymes may work in specific pH. We have enzymes that are found in the stomach that work in acidic pH. We have enzymes that are found in the mouth, where we have salivary amylase that works in slightly alkaline pH. So they work specifically in optimum pH that promotes their maximum activity. We have enzyme catalase found in living tissues, very common. In most cases, this is a very common kind of enzyme that is highly examinable. But how is the student able to write out their observation and give correlating deductions? Enzyme catalase can be observed in 3 different levels. We have observation using number of bubbles, colorless bubbles, where hydrogen peroxide as a substrate is broken down to give water and oxygen, which oxygen is going to be released off in form of colorless bubbles. Is the student able to see that the bubbles given off were few? Were they very many? And how does he deduce his answer? It can be in form of rate of reaction. Was the reaction very slow? Was it very rapid? Then at times, they may be told to measure the height of froth of those bubbles that are given off, the few bubbles and very many. Is the student able to see that the height of froth was maybe 2 centimeters, 1.8? They should be able to measure the height of froth to a maximum, which only stops at the brim of the test tube. Then we have enzyme catalase. We continue. Different conditions that may be examined on enzyme catalase. We've already talked about temperature and how it affects the enzyme activity. Substrate concentration. If substrate concentration is low compared to the enzyme, you find that the enzyme substrate molecule will readily be formed, and this reaction, in most cases, is going to be at a rapid rate. However, if the substrate concentration is high and the number of enzymes is low, the reverse is going to be true. What would cause the effect of substrate concentration to lower enzyme activity is when the substrate concentration is low. If there are fewer molecules of the substrate combining with very many enzymes that are readily available, you find that the reaction will probably take on the integer of getting slower. Surface area. How does surface area affect enzyme or rate of reaction? When we have a tissue, for example, I can take on a tissue of whole cube of Irish potato, 2 by 2. You cut the cube 2 by 2, and then we have a 2 by 2 cube, put it in a mortar and grind it. When we grind that cube and we have a whole cube and expose them to the same amount of hydrogen peroxide, you find that the one that is grinded is going to give off rapid effervescence, is going to give off very many bubbles in a short period of time. But the one which has been left whole, the whole cube, will give us moderate amount of bubbles, and its reaction will be of moderate kind of reaction. Enzyme specificity. Enzyme specificity. I can have change of substrate with a particular enzyme. If I have enzyme catalase and I change the substrate from hydrogen peroxide, by this time round, I change it to water. Both of them are colorless liquids. And of course, a student might not identify that they're any different. But when you change the substrate, do you expect the enzyme to work? The student should be able to explain why I changed from solution X, which is hydrogen peroxide, presented solution Y, and there's no reaction. There should be an explanation, meaning that enzymes are specific in nature and they only work on a specific substrate. We have cell physiology. Cell physiology can be examined using diffusion, which is movement of molecules from high concentration to low concentration. Diffusion may be studied in the lab using synthetic cell membrane, which is the whisking tube. For example, I may have a solution where I have mixed starch molecules mixed with glucose molecules. And we put this mixture in a whisking tube. After immersing this whisking tube in distilled water, you find that after 30 minutes, we draw off the water and test for these particular nutrients that were tied up in the whisking tube. You find that the reducing sugars, which is glucose, will be present after 30 minutes, maybe little reducing sugars present. When you boil that water after 30 minutes, it will turn from, boil it with Benedict, sorry, it will turn from blue and on boiling turn to pale green, showing that the reducing sugars have small molecules that are able to pass through the pores of the membrane into the water and hence are present after 30 minutes. We have osmosis. Osmosis can be studied using plant tissues. For example, and here we are looking at the cell losing water, the cell gaining water. For example, if I used a tissue of Commelina, I get Commelina and immerse it in a dilute solution, for example, distilled water. And then I get another piece of Commelina, which is 2 centimeters, and immerse it in a highly concentrated solution. After about 10 minutes, I look at the reaction. how are these two tissues behaving in these two different solutions of different concentrations? You find that the tissue of Commelina that has been put in a highly concentrated solution is going to lose water molecules by osmosis from its cells to the surrounding solution, causing the cells of this tissue to become flaccid. They lose water and become flaccid. And then the tissue will bend and lose height or lose length. Whereas the one that has been put in a dilute solution, like distilled water, is going to gain or absorb water by osmosis. This water is going to cause cell turgidity within the tissues. And then the cells, as they swell, they press against the cell wall, causing the tissue to become fully turgid. And this, the observation, you find that at the end of 30 minutes, these tissues are going to have different observation. The one put in the concentrated solution will appear to be soft, flabby, and easily bends. Whereas the one put in distilled water or a dilute solution is going to be rough, is going to be hard and stiff. It does not easily bend. Then we have physiology to do with soil components of soil, different types of soil, looking at their characteristics. How are they able to behave with water, water drainage, water retention, okay? And soil experiments, in most cases, when they're examining, they normally drill on soil drainage. For example, I can have 3 soil samples: clay soil, I'll have loam soil, and sand soil. Pour an equal amount of volume of water and try to see which type of soil is going to give me the first drop. Or else I can say, measure the amount of water collected in the measuring cylinder in the first 5 minutes and give a reason as to why. Clay soil has either none or 1 drop. Loam soil has moderate amount of water collected, while sand soil, almost all the water is going to pass through. So a student should be able to explain those phenomena depending on the type of soil presented, and then later apply to improvement of soil structure. Now, this application is where we come in, and it's where the new curriculum is going. We want the students to be in a format of problem solving. If the student has been presented with clay soil and loam soil, are they able to tell us how these 2 types of soils can be improved for crop production? How are they going to affect crop yields? And later you find that this can be addressed in the agriculture sector. Aims of biological specimen. When we're looking at biological specimen, we can have biological specimen from zoology. Those are the animal structures, feathers, bones, etc. And then botany, we can have plant structures, leaves, flowers, storage organs, etc. What is the aim of biological specimen? We normally present biological specimen to make accurate observations of the specimen and record those accurate observations, relate structures of the organisms to their function, compare organisms and structures of organisms, and even be able to manipulate different apparatus, equipment, and read off graduations skillfully by giving exact readings. Animal structures and organisms. Animal structures, we have commonly, students may be presented with arthropods. Arthropods can be either under class Insecta or class Arachnida. Can a student present identity? If I'm able to identify a cockroach as an insect, do I know the reason as to why I've identified it as a cockroach? The specific 3 reasons: has 3 main body parts, has 3 pairs of legs, and 3 thoracic segments. And go ahead, can they able to identify a spider, for example, a tick? as an arachnid and give reasons as to why you are identifying that spider as an arachnid. A student should also be able to give observable characteristics and state the structural characteristics that they have observed. If I've presented them with a cockroach, maybe I tell them to remove the hind leg, cut it off, observe it, state the structural characteristics. The student should be able to tell me how many segments of the leg they've observed. Are there special structures, for example, spines on the leg of the cockroach? Differences. Can they give differences between different arthropods? I give a termite, I give a cockroach, and maybe a housefly, and tell them to state the difference between the termite and the housefly. They should be able to see that the housefly has wings, while the soldier termite lacks wings. Then they should be able to see the texture of the body. The soldier termite is smooth, while cockroach is hairy. Dichotomous key. If they're presented with a number of arthropods, for example, we can present a cockroach, a soldier termite, a housefly, and any other arthropod. What are the rules of dichotomous key? What are those rules as an identification key? Is a student able to apply the rules? Dichotomous key is always one number less than the number of specimen provided. Dichotomous key has an alternative A and B on each number presented. Dichotomous key in any characteristic presented in any number above should not be repeated in the number below. So students should be having these rules on their fingertips. Drawings. When making drawings, what are the things we are looking out for? A student should be able to see that he has written the correct title that comes from the question itself. After writing a correct title, it will guide that student on the interpretation of what specific part of the specimen has been questioned. Is he able to make magnification? Labeling lines should be touching exactly what the student means to label. And then we have accuracy. In case the student has been told to draw only the head and includes the thorax, is that student accurate? The accuracy mark normally depicts whether the drawing passes or not. Any structures that are included in a drawing that is specific, you find that this drawing, in most cases, if leniency is applied, will only qualify for labeling marks and the rest will go, loses all. Adaptation. Is the student able to state how this organism is adapted to surviving in its natural habitat? What are those features that it has in its body that enables it to survive in its natural habitat and reproduce there? Can the student describe the habitat? We have mammalian structures, the teeth, bones. Can they identify with reasons? In bones, can the student try to describe the articulation of bones? For example, if we have the short bones, can the student be able to tell us which bone is articulating with the thoracic vertebrae? And what is the significance of the articulating bone? Even dichotomous key can be brought here. I can have the three short bones with a rib bone. Are they able to identify using the structural descriptions, drawings, adaptations too? Then we have animal structures. We continue, we have bird, feathers, bones, identifying them, giving differences between structures, stating the significance of the observed features, and also drawings. Fish, it can be whole fish. It can be the head of the fish that has the gills. It can be the tail, describing it, looking at how webbed. Why is the tail webbed? What is the significance of increasing surface area of the tail? And give us the ecological significance of these structural proportions and your orientation of scales, There's a time they brought students were supposed to map out a 2x2 drawing of scales, show the direction of scales, where they facing, and give the significance as to why scales are always facing towards the tail end and attached towards the anterior end. Then drawings can come as a whole structure or segmentation. Segmentation of structure is very common these days. I can present a student with a feather from birds, and I tell them to draw half of the structure, including the posterior end, or including the anterior end of the feather. So a student should be knowing the orientations differently. Is a student able to tell the posterior end of the structure, the anterior end, the lateral side, etc. Okay? And then we continue with animal structures. For the A level, we have dissections. Dissections, students should be able to observe internal and external anatomy of organisms, should be able to classify these organisms. The common organisms that are normally presented for dissection are, we have arthropods, the cockroach, we have amphibians, the toad, and mammal, where we can either vary from a rabbit to the rat. Is the student able to identify external features, the structures of the external anatomy, describe those features, describe their positioning on the whole body of the organism, and then even give us the significance of that structure to the organism. This can apply to, the external features can apply to the head, they can apply to the body trunk, visible structures on the ventral side of the organism. For example, why is the belly side of the arthropod lighter in color compared to the dorsal side? A student should be able to tell the significance of those colorations. Then we can also have the external features on the limbs. For example, the toad. Why are the limbs webbed? Why do they have jointed? Why are they jointed? Sex identification. Students should be able to identify the sex of the animal, giving reasons, okay? We have drawings. Here, the drawings are very many of different systems. It can be the alimentary canal and associated organs. It can be the nervous system. We can also have the circulatory system, how the different arteries and veins are going to interact with each other to deliver and draw off blood from the different major organs in the body of the organism. And even then, they should be able to state the role or describe the role or explain the role played by different parts of the body system. For example, when we look at the digestive system, the digestive system has associated organs. Do we know the role of the pancreas in the alimentary canal? Do we know the role of the salivary glands in the mouth? Okay, so they should be able to bring out those roles very well. Plant structures. Botany, we normally look at plant structures and organisms. These can be flowers, leaves, fruits, storage organs, and stems. Here we have drawings, identifying of reproductive organs in the flowers. Does the flower have both male and female? Giving reasons to identify. Leaves. And then we also have dichotomous key coming in there. Now I come to question tags. Sheila dwelled a lot on question tags, and I don't think I'll spend a lot of time here. However, our question tags in practicals, we are trying to understand the question tag in terms of words that are asking for information related to interrogating. We are interrogating. Related to observing. Is the student able to observe a certain feature and present his findings? Illustration, that is the drawings. Are you able to draw a particular part of an organism? Are you able to interpret the question and illustrate the particular part of content from a particular topic? We have question tags are action verbs. A student wants to know how best they can present their answer. As teachers, are we able to help out these naive brains? How best they can present their answers in practical format? The meaning of different question attributes in order to predict the best question interpretation. Let's help them out. Let's give them the question tags, understand them so that they can know the attributes that they need, the depth of the answer that is needed to predict the best interpretation of the question. Then we have common question tags in practicals. We have state. Here, the student just is required of simple recall, simple knowledge based on identity. That identity should be precise and specific on a specific part of content. Explain, to illustrate or elaborate the meaning of information, to relate observation and content. We have anatomical descriptive words relating function to a structure. Describe, data can be collected and recorded, and you should be able to describe, have the proper descriptive words. Then adaptation, I've drilled enough on this. However, the student should be able to relate structure to function, modification of that structure, or parts of that structure, more to fit the existence of the organism in particular conditions, particular environment for its survival. Then we have significance, very common. Is the student able to know the extent to which something matters? What is the importance of the structure to the organism? How certain factors or structures enable the organisms to survive? Compare. Compare is wide. The student should be able to give similarities at higher order thinking and differences between structures, and this should be precisely written. Comment. Comment is normally on test observation and interpretation skills. Here we are testing for how is the student able to observe a set of data. Is he able to interpret? Does he have interpretation skills? Then we go ahead to calculate. We can calculate surface area. We can calculate in graphs. We can calculate in very many, but the narrative here is to stimulate the student to think. Common mistakes made by students, wrong interpretation of questions. They normally make wrong comparisons, failure to precisely present results basing on certain biological principles, e.g., describe the level of activity of an active substance. The student should be able to see that at low temperature, enzyme catalase has a low rate of activity. Moderate activity by every 10 degrees increase in temperature, and then at 37 to 40 degrees Celsius, enzyme activity is at its maximum peak. Then we have a failure to differentiate content. For example, here, the most common I can use are flowers. Students are not able to differentiate between type of pollination, mode of pollination, and agent of pollination. They normally confuse mode of pollination with type. The type of pollination is either cross or self-pollination. Mode, the word mode means method by which, how is this pollination enabled? By wind, okay? It can be by an animal. Then the agent is the animal itself, is the water or wind. Inaccurate readings of apparatus, hence getting incorrect results. Wrong drawings, use of wrong specimen to answer questions. At times you present them with the particular specimen because it is hard for them to interpret. They just pick whatever is on the table that is easier for them and they present those answers. These are very common. as assessment is being done. Making wrong comparisons. Then we have setting skills for practical questions. When setting questions as teachers, the question should always be able to test a particular part of content, a particular order of thinking. Is it high order? Is it low order? Is a student able to state the difference between low order that just needs simple knowledge or recall, or high order? For example, the practical biological knowledge in terms of classification, differentiating structures, and adaptations. The question also should be able to test the observation skills of the learners, and hence utilize them to deliver a written interpretation. The test question should also test techniques of manipulating equipment. For example, is a student able to use a measuring cylinder very well? Can they read the graduation of a temperature on a thermometer, etc.? They should be able to test the skills of manipulating equipment that are used to study structures, e.g. use of microscopy when we're dealing with cytology, use of a hand lens. Can they tell the magnification using these two? Drawing skills. They also test drawing skills, making well-drawn drawings while considering correct indication of magnification. Are students able to calculate magnification? What is magnification? They should be able to deliver a correct title, is normally got from the question. Neatness. What are we looking out for neatness? Thin line, sharp pencil, and the line shouldn't be so thick. Then drawing. What are the drawing points? The student should be able to interpret the drawing and know what particular drawing points am I bringing out. For example, if we have a leaf drawn, draw specimen X is a leaf. Has he seen that this is a leaf that has a long leaf stalk or a short leaf stalk? How is the stalk? For example, if it is a commelina leaf, is the student able to see that there is a demarcation between the lamina and the leaf sheath? What is the detail of the leaf sheath and the lamina itself? Is it hairy or smooth? So all those details are looked into when we are bringing out those drawing points. Labeling lines. These labeling lines should touch exactly the structure the student means to label. Accuracy. When we go back to the feather I gave you as an example, if I've said the lower part of the feather, including the quill, is the student knowing where the part called quill is found? The learner should be able to interpret views. For example, we have dorsal view, that is the back side of a structure. Ventral view, the belly side of a structure. Dorsolateral, on the side, but on the back side. Posterior, that is the side that is towards the anal part. And then anterior is the side towards the head end. Questions also test delivery of a specific well-known phrase, instruction that is well understood by the learner. Learner should be able to produce a precise answer for every question that is presented to them with a sound reason. There should be, during exams or practical exams, I would really advise students to first begin with what they know best and then end with what they think they're not well conversant with. In conclusion, while testing these students, we need adequate and appropriate teaching and learning resources, especially through improvisation. At times we are hard up with specimen, but where need be, improvise for these practicals to be conducted well and for best results to be good. Then stimulate the learner's brain to think harder and deliver alternatives that are in the hot region, high order thinking region. Students should be able to define and explain difficult terms. Let's exercise flexibility and patience in order to adequately involve the learners in discovery and problem solving. Let's bring out the best in others, even when they know more than we do. Set to improve yourself, but not to prove yourself. For God and my country, I remain Arinaitu Catherine, a teacher of Mengo Senior School. Thank you very much.
Thank you so much, Madam Sheila. Sorry for that. Thank you so much, Madam Sheila Adnaitwe, for that elaborate presentation. Thank you so much. I hope we are going now to entertain the questions. But before that, Mr. Okelo Jime is back. Mr. Okelo Jime, is it possible to unmute?
Hello?
Yes, Mr. Okelo?
Are you getting me?
Yes, you're very clear now.
Okay.
Okay, now, Mr. Okelo, as you come in, one of the questions that I always find with the teachers is the thing of the abridged curriculum, because we know that this current senior force are the students who were affected most by the lockdown time. So I'd like to get a word from you or the assurances on the curriculum and how teachers ought to have gone about it, especially for these candidates who are going to sit for their exams this year.
Okay, thank you. Good evening, members. Thank you so much, Arthur, for the time and the organization. Thank you to FBA for the session of our teachers workshop. Thank you to, I remember, I think Mr. Dungu for his elaborate welcoming remark, and also to the host institution. Thanks, Sister Josephine. Sorry, I broke off a bit at a certain time. I did not follow the entire proceeding, but I would have loved to be throughout. Even now, I'm still on and off, but I will be in a position to share the later have with the teachers. One, concerning the bridge, you never will set according to the bridge. A bridge curriculum was given for the last year's candidate and this year's candidate. Therefore, we need to look out for how they were abridged according to their respective years, because they were affected differently. So the assessment will be in line with a bridge curriculum. There could have been possibly some gaps in some subjects, I think, in last years, but it doesn't mean that they are not being followed. Those are as a result of human error, but everything is in line with a bridge until when we reach a point whereby NCDC says there's no need for us to follow their bridge. Hello?
Okay, yes. Thank you so much, Jimmy, for the assurance.
Then secondly, maybe this is a general one.
Yes.
I don't know, there's